Hello students, in this module I'll be talking about the other two types of endosperms, namely the cellular endosperm and the helobial endosperm. Let us see how these develop. Look, listen and learn. So first let's understand what is cellular endosperm and how does this develop. So we all remember that second male gamete which is fusing with the secondary nucleus and forming this triploid structure which we now know is called as the primary endosperm nucleus. Remember, it is a triploid structure. Now let us see what happens here and how this development is different from the free nuclear development. Okay, just observe. Now what will happen here? Same way the nucleus will divide. Okay, but this time see what happens. So the nucleus divides and immediately it is followed by cell wall formation simultaneously. Does that make sense? So we can say here the nucleus is dividing and immediately cell wall is dividing, or you can say right from the beginning what you just saw was that the karyokinesis was followed by cytokinesis. Unlike free nuclear, where initially there was only free nuclear division and there was no cytokinesis, right? But in this case, what you see is karyokinesis is followed by cytokinesis right from the beginning. Does that make sense? And what you end up with is a cellular mass which you can call as the cellular endosperm. Remember that the endosperm development occurs in the cellular form right from the beginning. But this type of endosperm is not very common, okay? So you can say it is the less common type of endosperm, and this type of endosperm is specially seen in dicots. Is it okay? So this is the second type of endosperm called as cellular endosperm. Now let us look at the third type, which is called as helobial endosperm. And let us also see why it is called helobial. Okay, so let us see the picture again. What you are seeing again is the same picture where the second male gamete fuses with the secondary nucleus and forms this triploid structure which we all know is called as primary endosperm nucleus, or PEN. Let us see what happens here. Initially, what you will see is that single cell will divide and give you two nuclei. Is it okay? Now, the first division of the PEN will be followed by wall formation, something like this. Okay? What you just see here is the PEN divided and there's a big wall formation. So what you end up with basically are two chambers. A larger chamber at the bottom, which is also called as the micropylar chamber, and there's a smaller chamber on the top, which is also called as the chalazal chamber. Is it okay? Now what will happen? In both these chambers, free nuclear divisions will take place. Is it okay? Something like this. What you're seeing now is free nuclear division in both chambers. And how did the two chambers form? The two chambers formed by the cell wall formation right in the beginning. Does that make sense? So what you end up with is cell wall formation right in the beginning, followed by free nuclear divisions afterwards. Does that make sense? So it is something in between. So we can also call this as an intermediate endosperm development. Does that make sense? So what will happen in the end, you will have all the free nuclei divided in both the chambers, the smaller chalazal chamber and the larger micropylar chambers. Now it will be further followed by cell wall formation, something like this, again in centripetal manner, right? So what you are seeing now in both chambers, cell wall formation is taking place from out to in, or the centripetal manner, to ultimately give you a cellular mass which you call as the endosperm. Remember, it started with a cell wall formation in the beginning, then it had free nuclear divisions, and then it showed cell wall formation again, and that's why it is called as the intermediate type of endosperm. Now, why it is also called as helobial endosperm is because of the fact that it is specially observed in the series called as Helobiae found in monocots. Is it okay? So this was the third type of endosperm. So let us recollect what were the three basic types of endosperm based on how they developed. The first one was called as the free nuclear endosperm. The second one is called as the cellular endosperm. And the third one is called as the helobial endosperm. I'm just showing you what happens initially. Does that make sense? Remember, the nuclear endosperm is the most common type of endosperm. The cellular endosperm is less common and found mostly in dicots, and the helobial endosperm is found in the Helobiae series of monocots. Okay? So this brings us to the end of the module where I talked about cellular endosperm as well as I talked about the helobial endosperm. In the next module, I'll be talking about the development of the embryo. Till then, thank you and take care.